Welcome to the Cancer Genomics Cloud webinar for July. I'm Jared Rosowski, and I'm pleased to be here today as your guest host for the event. Our speaker today is Dr. Jolyn Rakita, a researcher that I've had the pleasure of working with for about 2 years now. Dr. Rakita will be speaking to us today about the OpenPedCan project. The data and resources for OpenPedCan have just been made available on the Cancer Genomics Cloud. One quick note about the run of show today: Dr. Rakita will present her work, and then we'll open up the floor for a group discussion about the resources and ideas to improve on making them useful and accessible to everyone. Please hold any questions you have until then, but feel free to put them in the chat if you want to make sure you don't forget them or make sure we address them later. At the end of the discussion, Dr. Sarah Fisher, who's a community engagement manager for the CGC, will give a demo of the resources and how to access them. Without further ado, I present our speaker. We're very pleased to have Dr. Jolyn Rakita here with us today. Dr. Rakita leads the Bioinformatics Translational Pediatric Oncology team at the Center for Data-Driven Discovery in Biomedicine at the Children's Hospital of Philadelphia. With a focus on mechanisms of RNA splicing that drive tumorigenesis and the identification of novel splice-derived neopeptides in pediatric brain tumors, her research also explores pathogenic germline risk variants and their influence on tumor development. Dr. Rakita is committed to advancing pediatric oncology through the collaboration and the development of scalable, open source analytical tools, frameworks, and data resources, utilizing platforms like Docker, GitHub, AWS, and Kavatica. Her pioneering work is transforming pediatric cancer research, offering new avenues for discovery and clinical application. The title of her talk is Accelerating Pediatric Cancer Research, Harnessing the Power of OpenPedCan Project. Take it away, Dr. Rakita.
Thanks so much, Jared, and thank you for the kind introduction, and thank you all for inviting me. I'm really happy to be here today to talk about some of our open source projects. So with that, I'll just go through an outline. I wanted to discuss today both the Open Pediatric Brain Tumor Atlas, or the OpenPBTA, which is sort of where this all started, and then our continuation of that project, the Open Pediatric Cancer, or OpenPedCan project, and then following that, one of our research applications that we've been, for which we've been utilizing this data in our own group. So just some background, a little bit of the history for the OpenPBTA, or the Open Pediatric Brain Tumor Atlas. It really started with the CBTTC, or the Children's Brain Tumor Tissue Consortium. This was launched in 2011. It was a consortium of multiple institutions. Currently, we have 32. It may even be more than that now. Institutions where we had global sharing of specimens for pediatric brain tumors, and along with clinical imaging and genomic data. So prior to the launch, there was really no centralized resource for the brain tumors. Couple that with the opening of the Gabriella Miller Kids First Data Resource Center, which happens to be at D3V, the Center for Data-Driven Discovery in Biomedicine, where I currently am working. We had sequenced the first set of pediatric brain tumors shortly before 2018, and released the first the data for the first 1,000 tumors in September 2018. This had gone through some of these initial Gabriella Miller Kids First workflows, and were released on Kavatica without embargo. So I just wanted to note later, the Children's Brain Tumor Tissue Consortium, CBTTC, became the CBTN, so I'll refer to it as the CBTN from then on. So I sort of joined D3B when all this data was becoming available, and was tasked with analyzing it. And so it sort of began as one crazy idea. I had known Dr. Casey Green from some other interactions, and he was at the time he was the director of the Childhood Cancer Data Lab at Alex's Lemonade Stand Foundation, and we talked on Twitter. I said, do you think that we can Twitter source people for help on a speedy pediatric brain tumor atlas landscape paper? He had just published a machine learning paper that they had written on GitHub using Manubot, and I was intrigued. And so I thought, could we do an analysis project in that same way? So he humored me, and so we started to brainstorm our requirements for the Open PBTA. We wanted it to be collaborative. We wanted as many people to join as were interested. We wanted the data to, it had to be publicly available so everybody could be using the same source data. It had to be harmonized. The metadata had to be organized. We wanted our analyses to be reproducible, and we wanted our analyses to be scalable in case people wanted to apply it to other data sets, larger data sets. So we utilized all of these tools that I'm showing on the right, and I'll go through each of these throughout the presentation. So a little bit about this Open PBTA cohort. It was initially just samples from the CBTN and the PNOC, which is the Pacific Pediatric Neuro-Oncology Consortium. These are all pediatric brain tumors. It was from 943 patients, just over 1,000 tumors. They went through these Kids First, Gabriella Miller pipelines, and the data is stored on Amazon and released in Cavatica. The middle shows some broad histologies that are included in this cohort. It's mostly made up of initial diagnostic tumors, and then on the right we have this broken down into cancer groups. And sorry for this being a little blurry, but these are some of the Kids First somatic workflows. We didn't release anything germline due to data access issues, so this was a fully somatic project. But what I'm showing in the pink boxes are the outputs from Kids First that we collectively put together as part of this data resource for the Open PBTA. So I think it's important to go through some of the GitHub repository structure just so that you're familiar with how these projects are set up. We have an analysis repository, and we have the same folder set up for this as with OpenPedCan. So we have first we have a Docker file which has all of the necessary requirements to run any analysis in the repository. So each individual can pull the Docker image and run any analysis that's on here. We have a download data bash script where the data can be pulled in release format, and this goes through an MD5 check. The data folder on the repository is empty, but when you download the data, all of the data will be placed into that data folder so that the analyses have the right path for that data. So this is what it looks like as you download the data. Once everything is downloaded, it'll go through an MD5 check. In case anything is wrong, you might see a failed here. So like I said, we provided the data in a versioned manner with release notes. For OpenPBTA, we ended at version 23 and then went on to OpenPedCan. So once we had the data and the repository set up, and we D3B partnered with the CCDL, we launched OpenPBTA in September 2019, which was Childhood Cancer Awareness Month. And this is a reference for our publication, but what we really aimed to do was to have this be a global collaborative open science initiative. We brought together researchers and clinicians from different institutions, universities, hospitals, and we characterized this first set of over 1,000 pediatric brain tumors. Through this project, we also created nearly 50 open source scalable modules to perform some basic cancer genomic analyses, and also provide over 100 ready to use thematic outputs. And we did this through using a pull request model and code review model for contributions. So this is a figure taken from the paper. What we would do is a contributor could propose an analysis by filing an issue, or we had pre-populated the repository with issues for analyses that we were interested in performing, and somebody could sign up for one. They would create a branch off of the main repository and then implement their analysis. And then simultaneously, we also wrote the manuscript in Nanobot. So in a different repository, the contributor could write their methods this went through pull requests and scientific and analytical code review. We also used Circle CI for our continuous integration model in OpenPedCan, wherein we have a training data set. So every file in the data release also went into a subset of that was created training data set so that upon pull request, that analysis would be run on this training data set to make sure that it passed. So it was a pretty collaborative project, and we pretty much for our group and for OpenPedCan use the exact same methodology. So one thing we learned through this process, since OpenPBTA was occurring right when Kids First was creating their somatic workflows, previously there were a lot of germline workflows being worked on for Kids First. We really worked closely with the engineers and iteratively assisted and benchmarked and validated these workflow outputs, which are shown here in red for Kids First. And then as we did this, we created files for release and then analysis modules in green, which also created files for release. So some examples are Kids First went as far as creating CNVs or SNV calls. And then within our project, we did consensus calling and filtering. We did mutational signatures. We created tumor mutation burden values for each sample. And so these are all separate analysis modules within OpenPBTA, and they created outputs which are available and ready to use per sample, or you can add on to those as you add your own data. In addition to those analysis modules, we created many molecular subtyping analysis modules. So OpenPBTA really enabled the molecular subtyping of pediatric brain tumors in the absence of methylation data, which is the gold standard for creating, for identifying which subtypes these tumors belong to. So in 2016, the World Health Organization created a summary of new integrated diagnoses, which include molecular data in addition to pathology data to create this integrated diagnosis. That was a major update for brain tumor diagnosis since 2007. So within the OpenPBTA, we used the 2016 WHO molecular subtypes, and were able to, without methylation data, create integrated diagnoses for 60% of these tumor samples simply by including key genomic features and performing some classification on RNA-seq for these tumors. We additionally added some low-grade glioma subtypes, which were implemented in this review in 2020. Not in 2016, but then some of these got put into this later 2021 classification. So a lot of this work was pretty fluid, and we sort of tried to do this in as real time as we could. And there were some samples where we, the molecular subtyping didn't agree with what the pathology diagnosis was. So we also went back and forth with clinicians and pathologists to review these samples, found a few misdiagnoses, some new diagnoses that made sense, but wouldn't have been caught through initial analysis several years ago. So all of these integrated diagnoses are available now in our histologies files in these data releases. So OpenPBTA also enabled the development of additional tools for the pediatric oncology community. One thing we were interested in doing, because we had two different fusion callers, Ariba and Star Fusion from Kids First. Ariba generates a lot of fusion calls, was prioritizing these for what's potentially oncogenic. Researchers don't really, how can we make life easier for researchers to go through these and see which ones are potentially actionable. So we developed a filtering and annotation workflow, which then we later decided to put into an R package and a Shiny application. This Anafuse R package was able to, we tested it on TCGA fusions. We had processed TCGA from the CGC and put that through our Kids First workflows. And using the gold call fusion set, we were able to retain 96% of those fusions. And then in a previous project that I was involved with during my postdoc, the NCI's Pediatric Preclinical Testing Consortium, we had these are PDX models. We had an acute lymphoblastic leukemia truth set, which was 27 and 23. I forget the total number. But these were samples that were PCR validated, had PCR validated fusions, and Anafuse was able to capture 100% of these as known putative oncogenic fusions. So we call that, we have a file in this data release called putative oncogenic fusions, and it goes through this filtering. So we also, for subtyping medulloblastomas, we utilized our transcriptomic expression classifier, which we had published either right before OpenPBTA came online or shortly after. So this is something that we tested on microarray and RNA-seq data. It was a gene expression ratio. We used highly expressed, differentially expressed gene expression ratios to distinguish these 4 medullo subtypes: Wnt, sonic hedgehog, group 3, and group 4. And across these 15 testing data sets, we're able to achieve an 85 to 100% accuracy for distinguishing the correct subtype based on what was in pathology reports for these subtypes. And here in A, you can see these are the t-SNE plots for RNA-seq, and then B for the microarray data. We think that these potentially these group 3s here that are clustering with group 4, it's possible that these are potentially even misdiagnoses, but this is what the data looks like. It's pretty clean. So we had used this for classification of medullos. And then we also did a little bit of science. This is one aspect of the paper where we had used this P53 classifier previously trained on TCGA data, and we ran it on our cohort and found an area under the curve of 86% here, accuracy for identifying P53 scoring. So the higher the score here indicates the more like dysfunctional, we'll say, P53. And what we did here is we looked at the P53 scores based on their altered status. So we also separately looked at mutation data to see whether or not they would be classified as activated, having one of these 2 amino acid changes, whether or not they might have loss based on a mutation in the DNA binding domain, a stop mutation. There were a lot of different categorizations we have written in the README as to what is considered a lost mutation, and then other were things that we just couldn't classify. And we found that both for samples that were deemed lost or activated, the RNA-seq values of the expression scores were very high. So we could predictably use RNA-seq to determine whether or not these samples had P53 dysfunction. I don't want to say lost because of the high scores we observed in the activated samples. So when we look at expression data, we also see that the activated samples do show this gain of function or higher expression in the activated samples versus those that have loss. This was actually, this led us to use the P53 scoring and status in some survival analyses. So we did this across many different subtypes, but we highlight in the paper this H3K28 mutant DMG or diffuse midline glioma sample tumor type, which is a lethal and aggressive pediatric brain tumor, high-grade glioma. And we found that P53 status actually results in a lower overall survival here in the green curve versus this orange curve. And then this is the forest plot showing the worse overall survival. The hazard ratio is higher than the reference of K28 alone. And we were going through this data at a similar time where we were retrospectively analyzing our PNOC cohort, PNOC 03 clinical trial, which was a DIPG clinical trial. And we found in that trial of only about 38 patients that P53 mutation did result in lower overall survival. And in addition, this mutation status, looking at BNC, was predictive of a poorer radiographic response to radiotherapy. So these tumors had a higher tumor volume post radiotherapy than samples that did not have P53 mutation, shown in box plot here too. So simultaneously, I feel like we're doing a lot of stuff simultaneously, but we also launched an effort, launched OpenPedCan with another department at Children's Hospital of Philadelphia, DBHI, the Department of Biomedical and Health Informatics. And we did this in support of the NCI's Childhood Cancer Data Initiative. And really what we did in collaboration with DBHI and Frederick National Laboratory was we were tasked with creating this pediatric molecular targets platform. And it really they wanted it to be a pan-cancer initiative. So what we did essentially was fork the OpenPBTA repo and started on adding data for other pediatric cancers. So in this data set, OpenPedCan V11 is where the MTP eventually stopped, but we included the target project, TCGA, this PBTA data, and then we have one data set from the Gabriella Miller Kids First data resource, neuroblastoma. We have some panel data, fusion panel data from the Division of Genome Diagnostics at CHOP. And this data is in OpenPedCan and also now in this molecular targets platform at NCI. To date, we have expanded OpenPedCan to version 15, and now we have data for over 6,000 pediatric cancer patients, about 2,800 are pediatric brain tumor patients. We do have metadata for 1,100 parents. As an aside, through another sequencing project, some parents of the germline of some of the PBTA patient parents were sequenced, and so we have those biospecimen IDs in there so that they're identifiable if people were analyzing that data and wanted to look at the somatic proband information, they would be able to link them in that way. We also have GTEx data, and then like I said before, TCGA. So I'm pointing out here some of the data that is available on the GDC, the target data, CPTAC, which I'll mention in the next slide. We have some proteomics data in here, and then TCGA. So the current state of OpenPedCan, we have integrated most of our multimodal brain tumor data to date. And then here in the pink are changes since OpenPBTA. I feel like there's not been that many years in between, but we have really tried to expand this as much as we could. We have proteomics data on here now. We have another CNV caller. We have methylation data processed. And then we have microRNA-seq for a small subset of the PBTA data, some more proteomics for adult tumors, RNA splicing, data. And then we have upgraded all of our molecular subtyping to be in line with the WHO 2021 subtypes for the pediatric brain tumors. So this data, or this, excuse me, we have a preprint for this on bioRxiv describing the data and the changes. Some of the changes are that the major changes that we lifted, we changed to GenCode 35, VEP 105, and we had to lift some of these adult tumor data and adult normal tissue data over. So the data isn't identical to what's in OpenPBTA because of the versioning, but we have some detailed methods available in bioRxiv that go through those differences. So now I just wanted to transition a little bit to share some of our work that we've done on RNA splicing and how we've integrated some of the OpenPedCan data into our own projects. And so RNA splicing, we may all be aware, can affect each hallmark of cancer. We analyzed splicing across pediatric brain tumors. I think we have about 1,400 pediatric brain tumors now that we are looking at initial tumor only across these different tumor types. And we find that of these different types of splice events, this single exon event was by far the most prevalent. We focus most of our paper, most of our project here on single exon events, inclusion or skipping events. On the right side, we are showing an upset R plot across all these different cancer groups, and do see that while we show recurrent splice events that are unique to histology, we do see a lot of overlap of events across different histologies. So we consider we consider these brain tumors to be pretty heterogeneous when it comes to splicing. We have also devised a metric called the splicing burden index, which we use to quantify the splicing burden. It allows us to quantify the splicing burden without a control set. So we do here a calculation of the splicing burden for each tumor compared to every other tumor. And we do see that it varies across different cancer groups, and that if you look at the plot on the right, this is the percentage of tumors within each cancer group that has either a low, which is the lower 25% of tumors, or a high SBI. So germ cell tumors, the majority of them have a high SBI versus low-grade gliomas, where just over half of them have a low SBI. We wanted to look at the relationship between splicing and tumor mutation burden. Tumor mutation burden, as I referred to it before, is a flat file that's available now as an analysis product from either one of our repositories. So we have TMB, we compared TMB and SBI here, splicing burden level across each of our cancer groups. And we find that I'm not showing it here, but if we analyze this across the whole cohort, there's a flat line of a relationship between tumor mutation burden and splicing burden. But when we start to look within cancer group, now you start to see a few differences where there's an inverse relationship between tumor mutation burden and splicing burden, but it's specific to histology. So I think one of the cautions that we've learned analyzing the splicing data is that you really have to make sure you're grouping your samples together properly, and that these may even change as we get into more nuanced subtyping. We next performed consensus clustering across the tumor histologies to see whether or not there were splice events driving any biology that overlapped different cancer groups. We found 12 clusters fit our data the best using an iterative analysis of over 200 combinations of clustering with different distance metrics. And across these 12 clusters, we saw that high-grade gliomas were by far the most heterogeneous, and they actually were in each cluster. So we focus our work on high-grade glioma specifically. We also, interestingly, found that this molecular subtypes belonging to each group. So in Medulla, I mentioned before, there were these 4 major subtypes, group 3 for sonic hedgehog and Wnt. I might have expected, because these are pretty expression-driven subtypes, that we might have seen all of sonic hedgehog in one cluster and all of Wnt in another cluster, but we really didn't see that at all, and nor for any of the other histologies where we looked at a subtype level. So this may indicate that splice-based clusters might be contributing additional mechanisms to tumor biology that are novel. We then did a cluster-based pathway analysis. We used gene set variation analysis here, and this is another module that we have in OpenPedCan, so it was pretty easy to use on the clusters. And focusing here on spliceosome, we sort of validated that those clusters with low GSVA scores 4, 3, 11, and 5 also had a lot of samples that made up the low SBI cohort versus clusters 1 and 12, which had a lot of samples that had high SBI. So that was a nice validation to see. And then we also took those GSVA scores for the spliceosome and correlated that with the SBI metric that we created and found a pretty nice tight correlation here. So we're pretty confident in using that SBI metric. We found that high-grade gliomas with a high SBI have aberrant regulation of splicing factors. So this is a high versus low SBI volcano plot. And we find that both the HNRNP and SRSF families were differentially expressed. And then in addition to looking at the RNA-seq, we looked at that CPTAC whole cell proteomics and do see that RNA and protein are correlating very nicely in our cohort. So we think that we can use these RNA data as surrogates for what might be going on with the protein. Next, we looked at recurrent splicing aberrations, and we really focused down on recurrent events that affect exons with functional sites defined by UniProt, and found that one of these exons with functional sites that was affected was a kinase, protein kinase CLIC1, which happens to be a regulator of splicing, and found that a lot of our high-grade gliomas have increased inclusion of this exon 4, which has that phosphorylation event, which here in the sashimi plots, you see a high exon 4 sample versus a sample with a low exon 4, just for reference. This exon event is positively correlated with CLIC1 expression. And in previous studies, this was found to be, these are the phosphorylation sites. In previous studies, that event has been found to be associated with increased expression. So it was nice to see here. And then also increased phosphorylation of the SRSF family of splicing factors. So we also looked at the DepMap, cancer cell line DepMap dependency atlas, and found that, again, if you look at the groups as a whole, you don't see a relationship, but when you look at that specific transcript that includes exon 4 and you split it in between, split the expression of the cell lines into those that have high inclusion or low inclusion, or high expression or low expression, those cell lines that had high CLIC1 expression for the transcript that includes exon 4 had lower CRISPR dependency scores, which means they were more dependent on that transcript for survival. So this only occurred in CNS and brain cell lines and myeloid brain lines. We then looked across all the brain tumor cell lines and picked KNS42 as one of the more dependent cell lines, and it was also happened to be from an adolescent for our next analyses. We performed, so we had the short read RNA-seq from Kids First, but then we performed long read Oxford nanopore RNA-seq on some of the CBTN cell lines and then KNS42 to just validate that we are seeing these inclusion levels in long read RNA-seq and that the short read wasn't an artifact. And then finally, we did some experimental work. We found a CLIC1 inhibitor, Sirtuvivant, and did a dose response to look at in KNS42 cell lines to look at whether or not there was an effect on proliferation. And here we see dose-dependent decrease in proliferation with higher doses of this inhibitor. And then using a cell titer glow cell viability assay, we also see a similar effect. This inhibitor is actually a pan-DYRK and CLIC1 inhibitor. So we also decided to do a morpholino knockdown experiment to specifically target that exon 4. And this is a qPCR here showing that we are wiping out that exon 4. 3 to 4 junction primers show that we are losing exon 4 expression, but we still have expression when we go from exon 3 to 5. And then that knockout of exon 4 reduces protein expression of CLIC1. And then we similarly did a cell titer glow with the morpholino morpholino infected cell lines and then see reduction in viability at 24, 72, and 96 hours. So following morpholino treatment, we also did an RNA-seq experiment. We have a lot of data that we're gonna show hopefully in this preprint. We'll have preprinted soon, but we looked at differentially spliced, differentially expressed, and dependent genes for the CLIC1 morpholino versus non-targeting morpholino. And when we look at, and these are in CBTN cell lines now, not the depth map, we look at CLIC1, these differentially expressed and differentially spliced from the morpholino experiment, we do see that CLIC1 is targeting some of the oncogenes that we have previously assayed in a CRISPR experiment to be dependent in some of these high-grade glioma cell lines. So we think that CLIC1 is a dependency in pediatric CNS tumors. We know from previous studies that this active CLIC results from exon 4 inclusion, which includes these 2 phosphorylation sites. We know from previous studies that it localizes these SR proteins to the nucleus and then cooperates to phosphorylate them and then induces mRNA splicing. So we think that CLIC1 might be mediating some of these changes via this mechanism. And so we're excited to keep exploring this data, but really wouldn't be possible without all of the data that we have from OpenPedCan. So we continue to grow this with as more multimodal data are being generated. And I just wanted to recap here before the demo that we have data available in GitHub. The analysis module code and outputs are available for reuse and use. And then we have full data releases via download script. This includes this, one of the most important files, I think, are the histologies file, which includes a lot of clinical and metadata, including all the molecular subtype information, survival, et cetera. It's all in this one file. And Cavatica public open access projects. So I also wrote here before, soon to be mirrored on the CGC, but they just came aboard the CGC as well, so that you'll get a demo on. And then we do have process data in PEDC Bio and the NCI's molecular targets platform. This is what the public projects look like on Cavatica, and then Sarah will show you in the CGC what they look like. So I have this open question after going through what I showed for OpenPedCan. Are there other ways in which OpenPedCan data can be provided and or presented to empower reuse by researchers? And with that, I wanted to acknowledge a lot of people and funding that got us where we are today. And a lot of these people from my team and then from CHOP bioinformatics have really been essential in making this work happen. So thank you.
Thank you so much. Can we go back to your open call slide?
Sure.
So I want to really encourage everybody present. Please let's, like, as a community, like, brainstorm a little bit. Dr. Rokita has shown us a lot of ways that this data can be reused. But does anybody else have any, like, ways that they would prefer to receive the data? We're going to wait a little bit for people to get brave enough. We have a question in the chat from Arco Sen. Do you provide sequencing files?
Do you mean BAM or which kind of files?
Yeah, Arco, yes. Arco means BAM or FASTQ probably.
So those are available because they're restricted access. You can request them either via dbGaP for the CBTN project, or you can go to cbtn.org and request access. They are all mirrored on Cavatica. They're in AWS, so they're easy to access. We do have MAF files available in the repository download.
Yeah. I wanted to sort of ask you a little bit about making these data files available for public access without having to do a data access request from dbGaP. You can't make the raw sequencing files available under those conditions, right? But can you talk a little bit about what kind? It's like derived data that you've got in OpenPedCan that people can work with, right?
Yeah, that's right. So it is all harmonized using the Kids First workflows, and we have links to all of the processing steps, the CWL workflows that were used for this exact data. But it is derived data, so it is a somatic MAF file. We don't have the germline data available, but it's all accessible via data access request. So we have copy number variants, SNVs, indels, fusions, structural variants, proteomics data.
Yeah, it seems to me that one of the like, I'm sure this is pretty obvious, but one of the like real values of having this data set available is that it is harmonized across multiple different projects. We have another user ask, or another attendee asking in chat, are VCF files available?
No, sorry. The VCF files are not available on OpenPedCan, but I believe we do have a public somatic VCF openly available in Kids First. It's just not in OpenPedCan because we use the MAF format there.
That's fair. And I think you can interconvert from MAF to VCF with some of the tools. We may even have those tools available on CGC. We have another question about how the genome sequencing was done. Hopefully not complete genomics, which I think probably means whole genome sequencing. And I think the answer is there's a variety of things is what you showed.
Yeah, so I think there is one complete genomics data set from Target, but we don't have that included here. Everything else is for whole genome. We have whole genome and whole exome, and there are some targeted panel data sets. And mostly, I think it was all Illumina. There's some BGI sequencing, which has a similar instrument to Illumina. So.
And then another question in the chat, are tumor histologies files available? I think you may have like touched on that briefly. It was one of your favorite files.
Yeah, yeah. I think it's the most critical file there is the histologies.tsv file. It's a biospecimen level file, and it has all of the metadata. So you can link that file to all the sequencing files via biospecimen, and then the diagnoses are there from pathology, and our integrated diagnoses and cancer group and molecular subtype are all there. There's also for brain tumors, we ran a methylation classifier once we did get the methylation data. So there are methylation classifications for those as well included in the histologies file.
All right. We have another chat that I think may not be something that you can directly answer because I think it has a lot of different answers. How do you go about adding more sequencing data to the repositories?
Sure. Yeah, so we've really done that through what we have available in Kids First or through that molecular targets platform project that we had with FNL. So as we bring data through our harmonization process, then we can add the data, but it's really been done on the D3B end. But if there are data sets that you're interested in adding, happy to hear what people are interested in adding.
Yeah, I think it might also be worth noting that the data that have been unified here are from major projects supported by the NIH and other organizations, where the data collection was all done in the same way. Information about the patients was all collected in the same way. So these are like the target data set, the TCGA data set. These are large sort of flagship projects. But having this derived data available for open access means that a researcher who has their own sequencing data that they want to analyze can actually co-analyze it with these derived files, can benchmark their analyses against what OpenPedCan has done. So, you know, if you have sequencing data that isn't in one of these sort of like big omnibus projects, you can still make use of this data by bringing your own data to it and seeing what you get out of that. And then we had one other question. I think the answer to this is yes. Are you considering RNA and DNA data integrated? Are you checking variants in RNA-seq as well? I think a lot of the exon splicing that you were talking about is RNA-seq, but did you do any sort of, have you done any sort of integration with germline variants with the RNA-seq?
Germline or?
I don't know if that question actually made sense.
So actually, I think I know what they may be asking. We did not actually run any RNA variant calling algorithms. I think that's something that's being discussed within the Kids First Data Resource Center, but that's not something that's a standard workflow right now, so we haven't done it. That would be something done upstream, and then we can do that comparative analysis downstream.
All right. I think that maybe my demo can help add a little bit of flavor to this discussion, because I can show everybody how you can access the data that the OpenPedCan project produced on the Cancer Genomics Cloud. So I'm going to shift to sharing my screen, and let me get to where I wanted to start from. So I think I can take over from you, and so everyone should shortly be seeing my screen. I'm logged into the Cancer Genomics Cloud. And what I want to show you is when you log in, where you can find this data hosted on the CGC and how you can bring it into your own projects. So if we're logged in on the main dashboard, we have this public projects in the header here. So when you click here, it takes you to the gallery of public projects, and two new ones that have been added very recently are OpenPedCan and OpenPBTA. And these are open access projects. So these projects contain all the files that you can work with and use without having to fill out a dbGaP data access request. And so the description on these projects explains where the data come from and directs you to the GitHub here for the OpenPedCan project, so you can learn more there. And then the release notes tell you for every new release that Dr. Rokita and her working group release, what has changed in the release. And some of this is that there have been files changed. Sometimes it is scripts. And then it lists all of the data files that are available in this release. So this is in the description of the project. In the files section of the project, there are two folders, and those folders will be the previous release folder containing all of the sort of versioned releases of this data set, and the version 15, which is the newest release. And the files are present here as they were listed in the description, and you can click on them and see what the different files are. I'm going to click on this bed file, which this looks like this is used in some of the analysis. It's an exome targeted regions bed file. So this gives the regions that are targeted by the TruSeq exome workflow, sequencing workflow. The way that you can get these files into your own project is you can either select them here and copy them out to a project that you may already have existing, or you can actually, from this public projects gallery, make a copy of the project for yourself. So you can click the copy project button. I usually like to give it a name so that I will remember where the project came from, and then select your billing group and click copy. And it will make your own personal copy of the data in your projects list. So that way you can work with it, you can include it in your analyses, whatever you want to do. The other thing that's really interesting and important about what Dr. Rokita has done here is having this GitHub repository. This GitHub repository versions all of the analyses so that the science is completely reproducible. And not only that, but if you wanted to participate in the OpenPedCan project, the way you would do that would be by working with the GitHub repository. So for example, there are in the analyses directory on GitHub, there are scripts available that represent the way that Dr. Rokita and her colleagues actually analyze the data to start with. So having the GitHub available to give like the versions of the data and give actual reproductions of how the analysis is done is super crucial for reproducible reproducible science, crucial for reproducible science. But I want to demonstrate to you guys an interesting way that you can use this GitHub. Oh, that one decided to pause. You can use this GitHub on the CGC in a data studio session. So I have opened a data studio session, RStudio from the CGC, and R has Git integration built in. And so RStudio in the cloud also has Git integration built in. And you can clone the whole GitHub repository by clicking New Project, Version Control, Git. And then all you need here is the repository URL. So you get that on GitHub by clicking code, and you just copy this HTTPS URL. You go back to RStudio and paste that URL into the repository URL field, and then you click Create Project. Now, this is a large repository, so as I click Create Project, we're going to find that it takes several minutes to actually begin cloning all of these objects in. But once it does so, and I had intended to be able to show you this in a live editor, but I can still show you in, I can't show you. My settings on this RStudio project have a timeout period of 30 minutes, which is a cost guards rail that we use on the CGC so that you don't accidentally walk away from a data studio and leave it running and rack up a lot of cloud costs. But, and we passed that timeout. So what you can see now is that all of those files from the repository are cloned here in my project. I can access the scripts in these analysis files. I think I wanted to look in particular at the chromosomal instability. And some of these are our markdown scripts, and so we can actually load those here and see that script like without even having to start the data studio environment. As I said, this is going to take a while, so I'm not going to finish this. But one of the nice things about RStudio and Git is that you can actually, when you open a Git project, you actually have a GUI, a graphical user interface for Git in RStudio that you can use to make pull requests if you wanted to, or interact with Git in whatever way you wanted to. So these resources are now available for everyone who is a CGC member. And this basically provides another way to access the work that Dr. Rokita and her colleagues have done, in addition to the other methods that Dr. Rokita mentioned earlier. I'm going to stop sharing my screen now, and I'm going to check the chat and see if there are any other questions or comments. I see that there was a question, what type of process somatic mutation data is available? And the answer to that was yes, SNPs, indels, scNAs, and SVs, plus fusions and RNA expression values. So I think it is 2:59, so right on the button. I think I will end our webinar here. Thank you so much, Dr. Rokita, for presenting. This was really wonderful.
Thank you so much for having me. And thanks for showing your demo, because I didn't know about using RStudio in that way either with Git. So that was useful.
I have never really yeah, thank you. All right.